Welcome back to the Deep Dive. Today we're tackling something, well, it sits right at this tricky intersection of global trade and protecting ecosystems. We're asking, is the main method we use worldwide to stop invasive species on ships? Is it actually, maybe, creating super invaders?
It sounds counterintuitive, doesn't it? But that's the core question.
Exactly. We're talking about ballast water. The water ships take on for stability. Historically, it's been like a free taxi service for organisms across oceans.
A really effective one, unfortunately. It's the #1 way non-native species get moved around the globe.
So the solution, mandated internationally, often involves chlorination, adding chlorine to kill things off. And our source material today is a really important paper looking right at this. It's titled, Can Chlorination of Ballast Water Reduce Biological Invasions? Lead author is Yiping Lin, published in the Journal of Applied Ecology back in 2019. And it's a deep dive into whether this chlorination process actually achieves what we hope it does in terms of risk.
And it's such a critical question because, well, global shipping is massive. Over 90% of commodities move this way. And like you said, ballast water is the main pathway for these non-indigenous species, NIS. It's leading to what scientists call global homogenization, basically. Everywhere starting to look the same biologically, which is not good for biodiversity.
Okay, so the authorities stepped in, the IMO, the International Maritime Organization, they brought in this D2 standard, which basically says ships have to treat their ballast water using systems like UV radiation or, very commonly, chlorination. The goal being, reduce the number of viable organisms getting dumped out.
Right, the intention is solid, reduce the numbers. But here is the potential flaw that Lin and the team zeroed in on. The D2 standard focuses almost entirely on something called total propagule pressure.
Okay, what's that exactly?
It's essentially the total abundance, the sheer number of living organisms being released. The researchers questioned if just cutting down the numbers really tackles the true invasion risk. They looked at another factor, colonization pressure.
So two different pressures. Propagule pressure is the count, colonization pressure is something else.
Exactly. And understanding the difference is key to this whole paper. Propagule pressure, the abundance, the numbers matters because, well, the more individuals you release, the higher the chance some will survive and establish. It helps overcome things like finding mates or just random bad luck wiping out a tiny group. Demographics, basically. Aliyah effects is the technical term.
Okay, so more is generally bad. Makes sense. But why is this other one, colonization pressure, maybe even more important? What is it measuring?
Colonization pressure measures the variety, the species richness, the number of different kinds of organisms being introduced. Think about it. If you release, say, 1 million individuals of species A, that's high propagule pressure. But what if you release only 1,000 individuals, each of 10 different species, species A through J?
Okay. Lower total number, maybe, but more. Shots on goal.
Precisely. More variety means a much higher probability that at least one of those species will find the new environment suitable, that it'll be pre-adapted, ready to thrive. So high colonization pressure, high species richness dramatically increases the odds of a successful invasion, even if the total number of organisms isn't overwhelming.
And the paper argues that the current IMO rules, focusing only on the total number,
They're largely overlooking this colonization pressure, the species richness aspect. If we're serious about stopping invasions, we need to know what the treatment, in this case, chlorine, does to the whole community, not just the overall head count.
Right. Killing 96% sounds great, but if the remaining 4% are the toughest, most adaptable species, that changes the risk profile completely.
That's the worry.
So how do they measure this? Especially the richness parts. You mentioned traditional methods, like microscopy, often miss things.
They did a really clever and quite ambitious field study. They used an actual working ship, a bulk carrier called the Federal Venture. It did 4 transatlantic trips, picking up ballast water in Canadian ports, some fresh water, some brackish. Voyages took between 8 and 19 days.
Wow, so real-world conditions.
Absolutely. Sailing all the way to Brazil. And they set it up carefully. They sampled water right at the start as an initial control. Then, at the end of the voyage, they sampled tanks that were just carried along untreated, that's the final control, to see the effect of the journey itself. And then, critically, they sampled tanks that had been treated with chlorine.
What kind of chlorine dose are we talking about?
They used standard industrial bleach, sodium hypochlorite, 2 levels, a lower dose of 10 milligrams per liter, and a higher dose of 20 milligrams per liter. Common levels in treatment systems.
Okay, but the real game changer wasn't the chlorine itself, you said. It was how they analyzed the samples afterwards.
Exactly. They moved beyond just trying to count critters under a microscope. which, as you hinted, is really hard. Think about it, tiny larvae, eggs, cysts, organisms damaged by the journey or the chlorine. Very difficult to identify accurately or even see sometimes.
So what was the alternative?
They embraced DNA metabarcoding. It's a high-throughput sequencing approach. Instead of looking for whole organisms, they extracted all the zooplankton DNA present in the water sample.
All of it, even from like fragments or dormant stages.
Pretty much anything with the target DNA marker. They specifically looked at a region called V4 of the small subunit ribosomal DNA, which is good for identifying different zooplankton groups. This lets you detect species even if they're present in low numbers or as eggs, or if they're too small or damaged to identify visually. It gives you a map of the entire community's genetic fingerprint.
That sounds incredibly powerful, like CSI for ballast water.
Kind of, yeah. The scale was huge. They processed something like 1.8 million raw DNA sequence reads. And from that, they identified and focused on 660 distinct zooplankton operational taxonomic units, OTUs. Think of OTUs as roughly equivalent to species for this purpose. This gave them unprecedented accuracy in measuring that key metric: colonization pressure or species richness.
Okay, so the setup is clear, the method is cutting edge. Let's get to the results. What did they find about species richness after the journey and the chlorine treatment?
This is probably the biggest headline finding. Neither the long sea voyage itself nor the chlorine treatment at either dose significantly reduced the colonization pressure.
Wait, really? The total number of different species, the OTUs, didn't go down?
Not significantly compared to the initial samples. The diversity indices, which are other ways of measuring richness and evenness, also showed no significant reduction.
But hang on, didn't earlier studies, maybe even on these same samples, show that chlorine does kill most things, that the total abundance drops massively?
Yes, absolutely, and the authors acknowledge that. Previous work, including studies by Paolucci and others in 2015 using these exact same voyage samples, confirmed chlorine achieved high kill rates. Total absolute abundance of zooplankton was strongly reduced, often hitting that 96% or higher mark required by regulations.
Okay, my head's spinning a bit. How can you kill 96% of the individuals, but not reduce the number of species present? Are some species just invincible to chlorine?
That's the crux of it. Chlorine wasn't acting like a broad-spectrum killer wiping out everything equally. It acted as a selective pressure. It didn't kill randomly across all species. It killed some species much more effectively than others. So while it didn't significantly lower the total number of species present, richness, colonization pressure, it dramatically shifted which species were most common among the survivors. It altered the community structure.
Ah, okay, so it's about relative abundance changing, measured by what they call relative propagule pressure.
Exactly. That's the proportion each species, or OTU, makes up of the remaining community after treatment.
So which groups got hammered by the chlorine?
The relative proportion of certain groups went down significantly, particularly things like mollusca snails, bivalves, that sort of thing, and rotifera, which are tiny wheel animals. Seems chlorine is pretty effective against them.
Okay, those were reduced. But that implies others survived, or even increased their share.
Increase their share, precisely. The relative propagule pressure actually went up for a couple of groups, specifically a group of protozoa called Oligohymenophoria, and also Copepoda, which are small crustaceans, a type of arthropod. These groups became proportionally more abundant after chlorination.
So the chlorine treatment is like filtering the community, removing the sensitive ones and leaving the tough ones behind.
That's a great way to put it. It's selection in action, and this has huge implications for invasion theory. If the treatment selectively removes the easy-to-kill species, the ones left over, these Oligohymenophoria and Copepoda, are, by definition, the ones that could withstand the chemical attack. They might be what biologists call exapted.
Exapted? What does that mean here? Like
Oh.
pre-adapted?
Kind of. It means they possess traits that evolved for some other reason. Maybe a thicker outer layer, maybe efficient toxin pumps that just happen to also provide resistance to chlorine. They weren't adapted to chlorine, but they had the right toolkit already. They passed a very tough environmental filter that the ship created. This suggests that these survivors, even if fewer in total number, might actually have a higher probability of establishing in the new port because they've already proven their resilience.
Wow. So the treatment itself could be selecting for future invaders? That's worrying.
It is. But the paper suggests another mechanism, possibly even more concerning, that could explain the increase in relative abundance of some groups.
What's that?
Dormant stages. Resting eggs, cysts, things like that. Many aquatic organisms produce these tough, dormant forms that can survive harsh conditions. They often settle in the sediment at the bottom of ballast tanks.
Okay, so they're just sitting there in the mud. How does chlorine affect them?
The researchers proposed that chlorine, particularly at that lower concentration they tested, the 10 mg dose, might actually act as a scarification agent.
Scarification, like scratching something.
Exactly. It could chemically scratch or weaken the tough outer shell of these dormant eggs or cysts. And this chemical stress, paradoxically, might actually trigger them to hatch.
You're kidding. So the chlorine kills the swimming adults, but wakes up the sleeping babies in the sediment.
That appears to be a strong possibility. It could explain why groups like Copepoda, which often have resting eggs, and some protozoa, which form cysts like the oligohymnophoria, actually saw their relative numbers increase after treatment. New individuals might be hatching out from the sediment because of the chlorine exposure.
That is deeply ironic. The cure potentially causing a new wave of the problem from a hidden source.
It highlights the complexity, and they found another interesting wrinkle related to this.
Which was?
This effect, the increase in relative abundance of tolerant, potentially hatching groups, was significantly more pronounced in the freshwater ballast samples compared to the brackish water samples.
Why would freshwater be different?
Well, producing dormant stages is a much more common life strategy for freshwater organisms. Freshwater environments tend to be less stable than marine ones. They dry up, freeze over, change temperature more drastically. So more species rely on dormancy. This finding suggests that freshwater ports, lakes, rivers connected to shipping routes, they might be facing an even greater risk from chlorinated ballast discharge than we thought because the treatment could be activating more dormant invaders originating from freshwater sources.
Okay, this DNA metabarcoding approach sounds revolutionary. But like any technique, it must have limitations, right? The authors mentioned some.
Yes, they were upfront about that. Science always has caveats. One key point is that DNA can persist for a while after an organism dies. So the DNA signal they detected could potentially include DNA from organisms the chlorine did kill. Dead organisms or just fragments, they argue based on other studies that this fraction is likely low, and their sample processing helps minimize it, but they can't completely rule it out.
Any others? What about just free-floating DNA in the water?
Right, eDNA, environmental DNA. That's also a potential confounder, though again, they use methods designed to focus on cellular material. There's also a technical challenge in high-throughput sequencing called tag jumping, where sequence reads can get wrongly assigned, potentially creating false positives for certain species. They use strategies to minimize this too, but it's an inherent possibility.
So even with these advanced tools, there's still some uncertainty in definitively saying these specific organisms were alive and kicking after treatment.
Exactly, which leads directly into their recommendations for the future.
What do they suggest? For management, for research?
For management, the big takeaway is that simply targeting total propagal pressure, that total abundance number dictated by the current IMO standard, is insufficient. Risk assessment absolutely has to consider changes in colonization, pressure, the species richness, and community composition as well. You need both pieces of the puzzle.
So the regulations themselves might need rethinking, moving beyond just a simple kill count target.
That's the implication. Focusing only on total numbers might give a false sense of security if the community structure shifts toward more resilient or potentially invasive species. And for future research, to address those limitations we just talked about.
Yes. How can we get better at telling lineys live from dead DNA?
They strongly recommend combining DNA analysis with RNA analysis. RNA, unlike DNA, degrades very quickly once an organism dies. So detecting species-specific RNA is a much stronger indicator that the organism was actually alive and metabolically active at the time of sampling. Combining DNA, who could be there, with RNA, who is likely alive there, would give a much more accurate picture of the viable invasion risk.
Okay, so let's try and synthesize this. The key message from Lynn and colleagues seems to be chlorine is effective at killing lots of organisms in ballast water, hitting those IMO abundance targets.
Yes, it reduces total propagal pressure significantly.
It's highly selective. It doesn't kill everything equally. It weeds out the weaker species.
Leaving behind a community potentially dominated by hardier, chlorine-tolerant survivors, the exapted ones.
And even worse, the chlorine itself, especially at lower doses, might be waking up dormant eggs and cysts resting in the tank sediment, potentially increasing the relative abundance of these tolerant groups.
Particularly for organisms originating from freshwater environments.
So the overall invasion risk, the actual chance of a new species establishing, might not be decreasing nearly as much as the simple organism count suggests. We might be trading high numbers of weak invaders for lower numbers of super invaders.
That's a stark but pretty accurate summary of the concern raised by this research.
So what's the path forward then, if chlorination alone has these potential downsides?
Well, it suggests we probably need to move towards more integrated treatment approaches. Relying on a single method, especially a chemical one that creates strong selective pressure, might not be enough. Perhaps combining chlorination with other methods like UV treatment or filtration or even heat could provide a more robust barrier that doesn't just select for one type of resistance. Multi-pronged systems.
Making it harder for any single species to have all the right defenses.
Exactly. The goal needs to shift slightly, perhaps, from just meeting a generic abundance number to truly minimizing the introduction risk of any potentially invasive species, considering their individual traits and resilience.
Which brings us to that final thought for you, the listener. Given that global trade isn't slowing down, and these ships are constantly moving water around, how do we balance the need for practical, cost-effective treatment systems like chlorination, which is relatively simple with the ecological imperatives to prevent invasions, especially when the treatment itself might be selecting for these highly resilient, exapted species?
Yeah, it's a tough balance. How much ecological risk are we willing to accept for cheaper shipping? Especially when the science shows our current main strategy might have these unintended consequences. Where do we draw that line?